I would really thank you the possibility for the organizers to to share with you some insights that we have been obtaining with some tools from our company. My name is Werner Rodriguez. I'm senior single cell solution architect from Becton Dickinson in the south of Europe. Today, we want to share with you some details and data that has been obtained, and we would really want to contribute to a better characterization of the immune cells identity of the immune cell systems, doing this with applying single cell analysis technology. The today example that I want to share with you is related with the recently non-innate lymphoid cells, and as we know, a part of the cytotoxic effect of the NK cells that are part of the innate lymphoid cells, there has been characterized into three major groups that are ILC1, ILC2, and ILC3. And as has been recently and deeply studied, these cells are mainly contributing to different reactions of our immune systems in case of inflammation, infection, metabolic disorders and allergies, and even in cancer disease. These cells are one thing that has been came out recently is also that they are not only different between among different groups. I mean, inside of these subsets that has been described before, but that they are even rare. So it's not always so easy to characterize them and to elucidate. One is because of their rarity, and second is because they are really, really heterogeneous, not only between different donors, but even between different tissues in the same donor. So this, for the study of these cells, represents, of course, a challenge. They are even sensitive also to manipulation, so we have to establish a very efficient method for the sample preparation, and some tools that will allow us to dig into this heterogeneity. We will talk today about cell sorting strategy for precise and effective analysis that will preserve the integrity of these samples and the even rare event that has been represented in our samples. We will see how the single cell multiomics approach would contribute with the simultaneous analysis of protein and gene expression. And then we will see how we can use this massive amount of data to create a better methodology of analysis with high parameter flow cytometry assays for a high throughput and for a characterization of large samples cohort. In a nutshell, what has been done is to collect samples from 3 different donors that has been treated with a reagent for the depletion of red blood cells, that is Eptoset. Then we split it, samples into one a liquid has been tested for a quality control assessment using a flow cytometry panel that we will see, and then another group of cells has been co-stained with 2 different types of antibodies. One is fluorinated for the study, has been able to use it into sorting procedure, and another one is antibodies conjugated with oligonucleotides, and we will see later how we can use it through molecular biology application to better characterize our samples. Then all the samples have been pulled together, and there has been sent through one unique sorting sessions in order to reduce the stress that could have been exposed to those samples. Then we have been apply a novel system for the isolation of single cells and to be able to monitor and to study in the same single cell at single cell level the expression of the mRNA. So we are talking about gene expression together with the expression of protein. So via APSIC analysis of this with these antibodies. And this allows us to create a library, to prepare a library that has been read into a sequencer, in this case in Illumina sequencing technology, and with this multidimensional data has been analyzed into a pipeline called 7 bridges, and then reanalyzed as a second round of analysis into a 6 gig software that allows us to dig into this data and to combine a very multimodal and multiparametric analysis, again, for protein and gene expression data. And then this allows us act as a base for developing a new and more specific panel for the flow cytometry at high parameter level. These are, in a nutshell, our panels that have been used for this analysis. In the extreme left, you will see the flow cytometry panel that has been used as a reference for QC control before starting the whole process. In the middle, we will see a sorting panel that has been used for sorting our cells, positively speaking, for CD127. And we wanted, of course, to eliminate what cells that would not be part of our ILC. And then we use for the molecular analysis, for molecular biology analysis, a panel of 42 plex APSIC antibodies conjugated with oligos, and as you can see, there are some specificities that are present in both panels for sortings and for the molecular biology analysis. This has been used for, well, first of all, has been demonstrated internally that there is no any compromise with the data that has been obtained with the core staining, and allows to optimize the workflow, and but also allow us to do a purity check via these antibodies conjugated with oligos, and we will see in the next slides how this has been working. So it is a very well-known technology or procedure is to double check our once that we sort our samples, is to double check that if our sorting has been done well. And one of these procedures is to resort our samples to make it go through the new process for sorting. But of course, this could be challenging with cells with with samples with a small amount of cells, and also with cells that are very sensitive to this process. So we take the advantage of the use of these specificities for the purity check that we have been included in our APSIC panel. This panel with antibodies conjugated with oligos, and we use it for the quality control. This allows us to gate specifically and to analyze specifically, to focus our analysis in what were the groups of cells that was really interesting for us. And as you see, this avoid us to eliminate a sample that otherwise would not be useful. It has been presented inside some specificities that were not as desired. So at the end, as you can see in the far right diagram, we were able to characterize the three major groups: ILC2, ILC1, and ILC progenitors, or ILC3. As a second quality control, we wanted to double check if the data that has been obtained from our assay with antibodies conjugated with oligos has been consistent, which has been observed with the flow cytometry. And as you can see, not only we did obtain the same proportion of representation of our cells, but also, as you can see in the column 1 with the donor 1, we have the approximately the same amount of data. As you can see, we have the same proportion of ILC2, ILC1, and ILC3 coming from the same sample. So what we have been see as a reference with flow cytometry has been even confirmed using APSIC panel. And going into the analysis of us has been done. Once that we obtain and combine together the data coming from the expression of a gene panel that is composed by 399 panel genes, related with the immune expression, and with the 42 antibodies that we saw before. What we did is a combination and a dimensional reduction and a visualization to allow us to see how every single cell has been grouped into different groups. And first, we has been used a manual gating using two specificities, CD 294 and CD 117, and this allows us to define our groups of cells. We apply this gating to the t-SNE plots that we see in the center and in the right side of the slide, and this allows us to make evident how these three groups of cells has been mapping in our population. But this was a kind of overriding of what has been seen, and but in the lower part of our slide, we are able to dig in and into focus of what is the specific expression level of expression of these proteins, as you can see. And we see that there are some expressions for the CD4, for example, in the ILC1 group, and as you can see, for example, in the CD103, it is only related with a specific subpopulation of ILC2. This data came from the possibility to have the ability to combine the differential protein expression with the gene expression at single cell level into the same experiment. As you can see here in this heat map, every column is one cell, and this allows us to have the vision and a glance of what is happening at not only at gene expression level, but also combining this data with expression of the proteins at a single cell level. In these slides, we can even see that we are able to correlate what is the level of expression of the gene and also the protein. And this is important. In that case, we wanted to even study what are the what is happening at a single cell level into processes that are occurring into post-transcription at the post-transcription level. So not only what is happening at the level of mRNA expression, but also at a level of expression of proteins. And again, you can see that it's clear, evident that there is one pattern of expression of CD4 into the ILC group that is not evident in the ILC2 and ILC3 group. And as a counterpart, we will see, for example, the expression of CD117 and mainly in the ILC3 group. This allows us to create and to begin more deep in what is the transcription and analysis of the ILC major subsets. And we saw that there are some positive expressions, for example, of the CD4 that is a classical marker for T cells. This is something that we have been even checking in the literature, and there are some controversial ideas about if they are a truly ILC type of cells, or there are some T cells that are contaminating these groups, and have to be elucidated what is the role of these type of cells that are actually included in the ILC group. Digging into our more into our data, as we can see here, there is also positivity for the CD4 and the CD8 in some cells. As you can see, this is more evident and only evident in our ILC group. So we have been even seeing what is also reported into literature. And as you can see, ILC2, ILC3 are lacking of these two markers. So at the end of this process, so one that we have been using all the data collected and analyzed in our SCI software has been used to better and to fine tune a specific high throughput, high parameter panel for the flow cytometry, and for that analysis, increasing on our sample cohorts, we have been used BDFX A56 cell analysis, cell analyzer. So what has been done is to create a flow cytometry panel using 21 colors, and there has been based in the biological information that has been revealed by single cell multiomics analysis. We then perform analysis for healthy donors using these subsets that allow us to begin what has been happening into ILC subsets, but mostly based in the information of the differential expression that has been obtained as a reference. And you can see here in the left part what is the source of our information. I mean, the combination of the APSIC expression and single cell RNA sequencing. And this allows to create these panels. As you can see, we were first divided into these three major groups using CD 294 and CD 117, and then in the lower part of our slide we see different histograms that evidently show the expression of the ILC1, the CD4 specificity for ILC1, and allow us to better characterize this new group of samples. And if we wanted to go in depth into the specific heterogeneity for ILC1, again, has been studied with bias flow cytometry using the specificities that we see again in the left side, and allow us to confirm what has been observe using the molecular biology workflow using the single cell analysis. So what we wanted to show you today is to a complete workflow and a total solution that allows us to better characterize one example of the effectors of the immune system that are in ion cells. You saw we were able to demonstrate the concordance between the flow data and the data coming from the molecular biology workflow with these experiments, and allows us to demonstrate the ability of the multiomics for the designing a high throughput panel for flow cytometry. With that, I would really thank you, and I will open now to your questions.